Hi everyone, welcome to the fifth episode of CSEC Biology with Mr. Charles. Today we are looking at the biology of cells, specifically the movement of substances through cells. Our learning objectives: by the end of this lesson you will be able to answer the following objectives. Explain what is meant by the terms diffusion, osmosis, and active transport. Outline the importance of diffusion, osmosis, and active transport in living organisms. Define tonicity. Differentiate between water concentration and solid concentration. Differentiate among hypertonic, isotonic, and hypotonic solutions. Discuss the effects of varying solid concentrations on cells of plant and animal tissues. Interpret results from osmosis experiments involving both potato and carrot cylinders in different solid concentrations, and explain simple osmosis experiments involving the whisking tube and Thiessen funnel. So let us look at the processes by which materials move into and out of cells. Okay. Substances move into and out of cells and from one cell to another by passive transport and active transport. Passive transport does not require energy. Active transport requires energy in order to occur. Passive transport includes diffusion and osmosis. Okay, so diffusion is the net movement of particles from an area of higher concentration to an area of lower concentration until the particles are evenly distributed. And these particles can be particles in gases, liquids, and solutions, right? All three are capable of diffusing. The particles are said to move down a concentration gradient, and it is important to note that diffusion is a way that cells obtain many of their requirements and get rid of the waste products or the waste products of cellular metabolism, which if not removed would poison them. Okay, the importance of diffusion in living organisms. Oxygen diffuses into organisms via gaseous exchange surfaces for use in cellular respiration. The waste product of cellular respiration, that is carbon dioxide, diffuses out of gaseous exchange surfaces. Carbon dioxide, also needed for photosynthesis, diffuses into plant cells, especially cells of leaves, okay, as a raw material. The waste product of photosynthesis, that is oxygen, diffuses out of leaves and plant cells into the environment. Some of the end products of digestion, some of the glucose and amino acids, diffuse through the cells of the ileum in the small intestine and capillary walls and into the bloodstream. Okay, so these are the importance of diffusion in living organisms. Now, osmosis is a special kind of diffusion. It is a movement of water molecules through a differentially permeable membrane from a solution containing a lot of water molecules, example a dilute solution or water itself, to a solution containing fewer water molecules, example a concentrated solution. A differentially permeable membrane has tiny holes which allow water molecules through. Sugar molecules would be too large to pass through the pores in the differentially permeable membrane. So here we have an example, a diagram showing us a differentially permeable membrane. On this side, the left side of the membrane, we have sugar and water molecules. On the right side, we have sugar and water molecules. Okay, the arrow tells us the direction of flow of water. So water molecules would pass through the pores, but sugar molecules would be too large to pass. Okay, the water molecules would go from where you have a little bit of solute to where you have a lot of solute. Okay, so from low solid concentration to high solid concentration, from high water concentration to low water concentration, because the ratio of water molecules to sugar molecules here is higher than the ratio of water molecules to sugar molecules here. So water would go from where it has a higher proportion to where it has a lower proportion through the differentially permeable membrane. Okay. Now, osmosis is important to living organisms just as diffusion. It is important to keep all cells hydrated, okay, by moving water through them. It is important, right, to keep cells turgid, and this is especially important, this causes non-woody stems to stand upright and keep them firm. Water is kept moving through plants by osmosis occurring in the cells of roots and leaves. This ensures that the leaves get water for photosynthesis. Okay, the size of the stomatal pores in the leaves, right, is regulated by osmosis occurring in the guard cells that surround those pores. This controls the loss of water from the leaves of plants. Okay, controls transpiration. Water is reabsorbed into the blood from the filtrate in the kidney tubules by osmosis, and this prevents the body from losing too much water. Okay. Now, when we are talking about water concentration, we are speaking of the proportion of a solution that is water. Solid concentration, the proportion of that solution that is solute, not water. So in every solution, there would be a water concentration and a solid concentration. There would be a percentage of the solution that is water and a percentage of the solution that is solute. Okay, a solution with a high water concentration is referred to as being dilute and has a low solid concentration. For example, if the water concentration is 70%, or in other words, if 70% of the solution is water, then 30% of that solution would be solute, or the solid concentration would be 30%. A solution with a low water concentration is referred to as being concentrated. Okay, low water concentration, concentrated solution. And this solution would have a high solid concentration, because if the proportion of water there is low, then the proportion of solids would be high. So in other words, if the water concentration is 30%, or if 30% of the solution is water, then 70% of the solution would be solid. So the solid concentration would be 70%. Then we have the tonicity of solutions. Tonicity is basically the concentration of a solution, right, compared to that of another solution. And when we are talking about concentration, we are talking about the amount of solutes that are dissolved in solution. Okay, a solution may be hypertonic, isotonic, or hypotonic. If a solution is hypertonic, then this solution is more concentrated than the one to which it is compared. If it is hypotonic, then it is less concentrated than the one to which it is compared. Okay, isotonic solutions both have the same concentration. Okay, if an animal cell is placed in a solution that is more concentrated than its cytoplasm, or if an animal cell is placed in a hypertonic solution, water would flow out of the cell by osmosis, causing the volume and the tugger pressure of fluid inside to be lower than outside the membrane, causing the cell to shrink and become shriveled or flaccid. Okay, so here some red blood cells were placed in a hypertonic solution, and the cytoplasm become shrinked, right, and the cell membrane become wrinkled and shriveled or flaccid, because more water or the net flow of water was from inside the cell to outside the cell. So the pressure inside the cell decreased, causing the cell membrane to wrinkle and become flaccid or shriveled. Okay, so that's the reason for the appearance of those cells here. Animal cells become wrinkled and shriveled only when they are placed in a hypertonic solution. However, if an animal cell is placed in a solution that is less concentrated than its cytoplasm, that is a hypotonic solution, water moves into the cell by osmosis. Okay, and this causes the volume and the tugger pressure to be higher inside than outside the cell membrane. Okay, as a result, the cell would expand and become firm or turgid. And if water continues to enter the cell, the pressure exerted on the cell membrane internally would increase, and this would eventually cause the cell to burst or undergo cytolysis. And this is what you're seeing happening here. These red blood cells were placed in a hypotonic solution, and because the concentration inside was higher than the concentration outside, or the concentration outside was lower than the concentration inside, there was more water molecules on the outside, so the water flowed from outside the cells to inside the cells, increasing the volume and pressure inside there, and since the cell membrane cannot withstand too much pressure, it would burst. Okay, and the cells would undergo cytolysis. It would explode and its contents would be extruded into the external environment. Now, if an animal cell is placed in a solution of the same concentration as its cytoplasm, that is an isotonic solution, the rates at which water molecules enter and leave the cell would be equal. Okay, the volume and tug pressure on both sides of the cell membrane would be the same, and as a result, the size of the cell would be also maintained, or would also be maintained. And that is why the cells here in the middle here have normal shapes and sizes, because they were placed in an isotonic solution, so the amount of water entering was equal to the amount of water leaving the cell. Okay, and that is what happens in an isotonic solution. Okay, most of our body fluids are isotonic, right? That's why our cells are able to maintain their normal shapes. Now, if a plant cell is placed in a solution that is more concentrated than its cytoplasm, that is a hypertonic solution, water would flow out of the cell by osmosis. The volume and tug pressure inside the membrane would be lower than the volume and pressure outside the membrane. And because of that, the cell membrane would be separated from the cell wall, right? Causing the cell to become plasmolized, or plasmolysis to occur. Okay, that is what happened here. That is the reason why the cell membrane detached from the cell wall, because the pressure inside was not enough to push the cell membrane against the cell wall. Right, that pressure inside the membrane is created by the fluid inside the membranes, and when water leaves the cell, the pressure decreases. So the likelihood of the cell membrane attaching itself to the cell wall decreases. Okay, and then the cell become plasmolized. Okay, and that is what happens when a plant cell is placed in a hypertonic solution. Now, if a plant cell, on the other hand, is placed in a solution that is less concentrated than its cytoplasm, that is a hypotonic solution, water would move out of that cell by osmosis. The volume and tug pressure would be higher inside than outside the cell membrane. The cell would expand and become turgid. Okay, now due to the rigidity of the cell wall, a plant cell would not burst under tug pressure because of that rigidity there. So here we have a plant cell in a hypotonic solution. Though water is entering the cell and the pressure inside is increasing, the cell would not burst because of that rigid cell wall structure. Okay, unlike the animal cell, right, the cell membrane is not able to withstand the pressure inside there, so it would burst under the increased pressure in a hypotonic solution. But in a plant cell do not undergo cytolysis because of the cell wall. Okay, that gives the borders of the cell added strength and rigidity. So remember, if an animal cell is placed in an isotonic solution, it would maintain its shape and structure because the amount of water entering is the same as the amount of water leaving the cell. Same thing with a plant cell. The amount of water entering would be the same as the amount of water leaving in an isotonic solution, so the plant cell would maintain its shape and structure. In a hypertonic solution, however, more water would be leaving the cells than entering. And as a result, you would have a net loss of water from the cell, and the cell would lose pressure and volume on the inside, causing the animal cell to become shriveled and wrinkled, okay, or flaccid, and the plant cell to become plasmolized as the cell membrane separates from the cell wall. In a hypotonic solution, the animal cell would expand, okay, and become turgid. And if water continues to enter the cell, then it would eventually burst under the pressure inside because the cell membrane cannot withstand so much pressure. However, though water is entering the plant cell in a hypotonic solution, okay, the cell wall protects the cell from exploding or from undergoing cytolysis because it is very rigid in structure. So the plant cell will just be a normal turgid cell. Okay, it would not explode. Plant cells resist cytolysis. So here we have 5 test tubes containing 5 different concentrations of salt sugar solutions with 5 cylinders, that is potato cylinders in them. These potato cylinders have the same mass and length, and the concentration of sugar solution increases from left to right. The concentration of sugar solution increases from left to right. So here we have a 0.1 molar solution, meaning that, right, for every 10 molecules of water, you have 1 molecule of sugar. Here, for every 10 molecule of water, you have 2 molecules of sugar, right? Here you have 3 molecules of sugar in 10 molecules of water, 4 molecules of sugar in 10, 5 molecules of sugar in 10. So as you go from left to right, the amount of solute in solution increases. The amount of sugar in the water that is dissolved in the water increases. Okay, and since the amount of solute increase, water would have been used to dissolve the solute. So the more solute you have in a solution, the more water you use to dissolve them. Okay, and as a result, less water would be available to dissolve more solutes. So as you go from left to right, the amount of water or the water concentration decreases and the solute concentration increases. Okay, now let us look at the table. Okay, here we have the test tubes. We have the concentration of sugar solution here from 0.1 to 0.5. Here we have the initial masses of potato cylinders in grams. All of them had the same mass. Okay, in this experiment, the length was not measured, but most of the time, all of them are of the same length also. The final masses were determined after the experiment. Notice that each of them is different, right? And each of them is different to the initial mass. And here we have the change of mass. Here we have the percentage change of mass of the potato cylinders. Okay, so for the cylinder in test tube A, notice that it was placed in the 0.1 molar solution. Molar is the unit we use to measure concentration. It means mole per liter or mole per cubic decimeter. Okay, it is what we use to measure the concentration of a solution. So here for A, after osmosis would have occurred, okay, the mass of this cylinder here increased from 2.0 to 2.2 grams. So we had a 0.2 increase in mass because 2.2 subtract 2.0 is 0.2. So we had a 2.2 increase. So the plus sign in front of the 0.2 means increase. So we had a 0.2 increase. Okay, is 2.2 subtract 2.0 is 0.2. So we had a 0.2 increase, or 0.2 gram increase in mass. Okay, so it's expected that this cylinder would get taller by 0.2 grams. Okay, or not necessarily taller by 0.2 grams, but taller because it could get wider also. But if you take the 0.2 and you divide it by the initial mass, you would get the percentage increase in mass. So 0.2 divided by 2.0, and you could try it on a calculator. Let me pull up a calculator here. Okay, let me try pulling up a calculator. Okay, let's calculate these things. So we have 0.2 divided by 2.0, you get 0.1. Multiply that by 100% and you get 10%. So here we have a 10% increase in mass. for this first cylinder here. Okay, for this second cylinder, we had a 0.1 gram increase. So 0.1 divided by, okay, 0.1 divided by 2.0, which was the initial mass, equals 0.05, and 0.05 times 100% would give us 5%. So we get a 5% increase here. Okay, you always divide the change by the initial to get the percentage change. Okay, here the initial mass was 2.0 grams, the final mass was 1.8 grams. So notice that for the third cylinder, actually the third, fourth, and fifth cylinder, you had decreases in mass, right? Because the final masses are lower than the initial masses. Okay, so to get a change in mass, you have to say final mass subtract initial mass. That will give you the change in mass. Okay, so final mass subtract initial mass, 1.7 subtract 2.0, you get 0.3. okay, 0.3. That's for this cylinder here, the fourth one. And if you take the 0.3 and you divide it by 2.0, which is the initial mass, right? Negative 0.3, sorry. So 1.7 subtract 2.0, right? You're subtracting a larger number from a smaller number, so you get a negative number, negative 0.3, and that means that the mass of the cylinder would have decreased. That's what the negative sign means. The mass of the cylinder would have decreased. Okay, and if you take that negative 0.3, okay, negative 0.3, and divide it by, okay, let me just clear that. So 1.7 subtract 2.0 is negative 0.3 divided by 2.0, which is the initial. You get negative 0.15, and then when you multiply that by 100, you get negative 15%. So you had a 15% decrease in mass. Here you had a 25% decrease in mass. You had a 10% decrease in mass. Okay, so let us really analyze this thing. Oh, by the way, look at the formula here. Okay, so for the change in mass of the cylinders is the difference between the initial mass and the final mass. So you take the final mass and subtract the initial mass from it to get a percentage change. You take what you get here and divide by the initial mass, multiply by 100% to get the percentage change. Okay, change could be increase or decrease. That's why you have the plus sign for increase here, the negative sign for decrease. So the masses of the cylinders in the test tubes A and B increase. So if you notice that both the masses for the change in mass for A and B had a positive sign in front of each of them, that means that there was an increase in mass, okay, in the cylinders, and a percentage increase in mass. This is because the solutions were hypotonic compared to the concentration of their respective cytoplasms. Okay, so the 0.1 molar solution and the 0.2 molar solution were each hypotonic, right, compared to the solutions of their respective cell cytoplasm in the potato tissue. Okay, so because the concentration outside the cell were lower than the concentration inside the cell, you had more water outside than inside. So water moved from where you had more of it to where you have less of it. Or another way you could say is that the solid concentration outside was lower than the solid concentration inside. So water moved from where you have less solid to where you have the higher percentage of solid. Okay, so that is why A and B had an increase in mass, or a percentage increase in mass. Okay, so that's the reason there. Now the cylinders in tubes C, D, and E had decreased mass, and this is because the solutions were hypertonic. So A and B were hypotonic, C, D, and E were hypertonic. And this compared to the hypertonic compared to the concentration of the respective cytoplasm. So you had more solid inside, so you had more solid outside than inside. So because you had more solid outside, you had less water outside. So wherever you have more solid, you would have less water. So water would move from where you have less solid or more water to where you have more water, where you have less water and more solid. So water move from inside the cells of the potato cylinders to outside. And because remember, the water inside the cells contributed to the initial mass. So if water leave the cells, then they are losing mass, and actually that type of mass they are losing is actually wet mass. Okay, so they are losing wet mass, and that would cause their mass to decrease. That is why for these three cylinders you had percentage decreases in mass. Okay, because water left the cells, right, the cells of the cylinders, and enter the solution or the hypertonic solution, causing the mass of the cells to decrease. Now it is expected that this cylinder here at the end of the experiment would be the tallest, and this one here would be the shortest, because this one lose the most water, this one gain the most water. Okay, so this one here would be second tallest, then this one, then this one, then this one will be the shortest, because as you move from left to right, the volume of water or the proportion of water in solution would decrease, okay, and the proportion of solid would increase. And wherever the proportion of solid in solution is highest, the cells would lose the most water. Now it's important to note that none of the solutions were isotonic. Okay, none of the solutions had the same concentration as the cell cytoplasms, and this is evident because none of these values here is 0. Okay, if there was an isotonic solution, then one of these changes or percentage, these changes in mass or percentage changes in mass would have been 0. Okay, now but by inference, or by way of inference or deduction, now we would actually expect that an isotonic solution would be between the 0.2 molar and 0.3 molar solutions. Okay, so somewhere between those two solutions, so maybe 0.2 something, right? 0.2 something molar would be an isotonic solution. So that's actually the approximate concentration of the cell cytoplasm, somewhere between 0.2 and 0.3 molar solution. Okay, and that's by inference. Now what I want you to take away from this slide is that as the concentration of solutions increases, the percentage increase in mass of the cylinders decreases, and this is because the volume of water outside the membrane of each cell also decreases, causing the ease with which water enters the cell to decrease. Okay, so as you move from left to right, the concentration or the solid concentration increases. Okay, and as the solid concentration increases, okay, most time when we are talking about concentration, we are talking about solid concentration. And as the solid concentration increases, okay, the likelihood that water would enter the cell decreases. Okay, that is why as you move from left to right, the percentage change in mass decrease, right? Decrease linearly because the concentration increase linearly. So it's an inverse proportionality relationship, an inverse proportion relationship. Okay, good. The same thing, the same experiment can be conducted with carrot cylinders. So here we have 5 test tubes, okay, with 5 carrot cylinders in 5 different concentration of salt solutions. Okay, so it's important that you have the same amount of solution in each test tube. Okay, the only thing that should change is the concentration of the solution. Everything else should remain the same. Okay, everything else should remain the same. The temperature of the solution should be the same. The length of the cylinders should be the same. The mass of the cylinder should be the same. The only thing that should change is the concentration from left to right. So this is least concentrated or most dilute. This is most concentrated or least dilute. So least concentrated here because it has the least amount of solid, most dilute because it has the most amount of water. A 0.1 molar salt solution would have 10% salt, and let me show you the calculation. Okay, so a 0.1 molar solution would have 10% salt. Okay, 0.1 molar salt solution, and you would know that by multiplying the 0.1 by 100. Okay, so you get 10% of the solution is salt, and if 10% is salt, then you could find what percentage is water. So, and you could find that by saying 100 subtract 10, that would give you 90. So 90% of the solution is water. So this is in 90%. The water concentration is 90% here. Right? The water concentration here is 80%. The water concentration here is 70%, 60%, 50%. So as you move from left to right, the water concentration decreases. Okay? So since the water concentration decreases from left to right, provided that other factors are kept constant, like other factors meaning the length of the cylinders, the mass of the cylinders, the amount of solution in the test tube, once those are not changed, then as you move from left to right, since the water concentration decreases from left to right, the likelihood of water entering the cells would decrease, right? Because here you have the most water in solution, so most likely water would enter the cells. Okay, here you have 80% water, so water is likely to enter the cells also. But from here to here, okay, the water start going down and going down, so the likelihood of water entering the cell would decrease. Water would tend to more exit the cell, so you would more have a flow of water out of the cells and inside of the cells because you have less water here. This cell here, of all the cells that lose water, okay, of all the cells that lose water, of all the cylinders that lose water, this one would lose the most water because it is in a solution that has the least water. Okay, so water would flow out of the cell. So the same trend, as the concentration of solution increases from 0.1 molar to 0.5 molar salt solutions, the ease with which water moves into the cells by osmosis decreases. Okay, so it is less likely for water to enter the cell as you move from left to right, or from least concentrated to most concentrated, right? And I'm saying from least concentrated to most concentrated because it doesn't always have to be arranged in that order. So for instance, it could have been 0.5 molar, 0.4, 0.3, 0.2, 0.1. Okay, it doesn't matter how you arrange the test tube once they are in order, but once they are in ascending order, right, the likelihood of water entering the cell would decrease. Now, it is also expected that the length of the cylinders after the experiment would decrease from left to right because this cell here, this cylinder here would lose the most water. And because it is losing the most water at the end of the experiment, it is expected to be the shortest. This one here is gaining the most water, so at the end of the experiment, it should be the tallest. So you should have this gradient of height from tallest to shortest, right, after the experiment. So this one will be the tallest, then this one, then this one, then this one, then this one will be the shortest because water is lost, right, in that order. Okay, the mass of the cells would also decrease in that order. Okay, so this one would be the heaviest, then this one, then this one, then this one, and this one. Okay, so keep those in mind. Now, we also have the whisking tube experiments, and there are two models of the whisking tube experiments I will show you because I do not want to go too much in depth with it. All right, two simple models. what actually is a whisking tube? It's an artificial differentially permeable membrane. Okay, and as we said before, a differentially permeable membrane is one that has pores in it that allows water molecules to pass through. Okay, or small molecules like water to pass through. Now, the differentially permeable membrane is also found in the whisking tube, so we can actually artificially create a differentially permeable membrane, and that is evident by the fact that we have one in the whisking tube. Okay, the whisking tube is also called a dialysis tube. Okay, or a dialysis tube. Here, this is a whisking tube. Notice that it is tied tightly here to prevent the solution inside from escaping into the solution outside in the beaker, and that is what escaping into the water in the beaker. And on this end, it is tied to a capillary tube. A capillary tube is basically a thin, long, tube. It has a very narrow or an extremely narrow diameter, okay, or extremely narrow cross section. So it's tied to a capillary tube here, and it's tied here, okay. So the solution inside here cannot escape easily. It can only pass. You can only have transfer between the holes of the membrane, right? And it's only the water molecules that would cross. The sugar molecules cannot cross because they are too large, okay. So you should not have sugar molecules inside here at the end of the experiment, because these two ends supposed to be very tight, okay. So inside the whisking tube we have sucrose solution. Okay, we have sucrose in solution. Here we have pure water. So the solid concentration inside here is higher than outside here. So outside you have more water molecules, right? The concentration of water outside is higher than the concentration of water inside. So water would move from outside to inside, and since water is moving into the whisking tube bag, okay, the level of the sucrose solution in the capillary tube would rise, okay, and that is evidence that osmosis would have occurred. And osmosis only occurred there because you had a gradient in the concentration of solids and the concentration of water from one side of the membrane to the next, okay. Remember, it's a gradient that causes osmosis to occur. Just as the gradient allows diffusion to occur, it's a gradient that allows osmosis to occur, right? From highest to lowest. Good. So highest water concentration to lowest water concentration, okay? That is where water would move, in that direction, okay? Now, let's look at another whisking tube experiment. Here we have this whisking tube in a beaker of water, in a container of water, okay. It's tied here, okay, just to ensure that you get a fair test and water is not leaving through any unauthorized exits, okay. So here we have water in the beaker. We also have water inside the whisking tube bag, and this water inside here is of the same concentration as the water outside here. So we have a we have an isotonic solution. Isotonic, the concentration of both solutions are the same. So this is a control experiment, and at the end of this experiment, it was observed that the whisking tube bag had the same mass, okay, and it retained its mass because there was no net flow of water across the membrane. And no net flow does not mean that the water did not pass the membrane. It just means that the rate at which water entered equals the rate at which water left. So the same amount of water that entered the membrane was the same amount of water that left, and because of that, there was no net flow of water across the membrane, and the mass of the whisking tube remained the same at the end of the experiment. However, here, where we actually had 60% sucrose on the inside and 0% sucrose on the outside, we have a gradient. So high solid concentration inside, low solid concentration outside, or high water concentration outside, low water concentration inside, water would always move in the direction where you have the higher percentage of solute. So water moved into the whisking tube bag, increasing the turgidity of the bag and increasing the mass of the whisking tube bag at the end of the experiment. Okay. Now here we have the thistle funnel experiment. So here at the start of the experiment, or in the beginning, we have 10% solute inside here. So this is a 10% solution, 10% solid, 90% water. Outside here we have 5% solid, 95% water. Okay. Now water would cross a membrane that would be beneath the thistle funnel. So there would be a membrane beneath the thistle funnel that is differentially permeable. So it would allow water molecules to pass through, right? Up and down. Because in the direction of the gradient. Okay, so here we actually see what is happening. So this is a membrane beneath the thistle funnel. This is the differentially permeable membrane here in pink. Notice water is flowing in both directions, but you have more water going up into the funnel than coming down from the funnel. So you have a net movement of water inside the thistle funnel. Okay, and notice also it's only the water that moves and not the solid. So you have a net flow of water into the funnel. Okay, and the water flows in the direction where you have the most solid or the higher percentage of solid. Okay, or the lower percentage of water. Here you have a high percentage of water and a low percentage of solid. Here you have a low percentage of water and a high percentage of solid. So water would move upwards into the funnel. And because water is moving into the funnel, the solution in the funnel would rise. Okay, it would rise due to the movement of water towards a higher percentage of solid. Remember, the higher percentage of solid was inside the funnel. Okay, so water moved in the direction of the higher percentage of solid, causing the level of water in the funnel to increase. Right, so osmosis, you have to give it a lot of time, right? Notice I have dedicated 12 slides to osmosis. Okay, right from slide 5, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, right? 12 slides for osmosis. So you have to give osmosis a lot of time, okay? Because it's very intricate detail. You have to read and pay attention and focus on the content, listen to the video over and over again to grasp the concept. So let us move on to active transport and its importance in living organisms. So active transport is a movement of particles through a membrane or through membranes against their concentration gradients. Okay, so you need energy to do that. Energy generated during respiration in the form of ATP is used to transport particles from an area of lower concentration to an area of higher concentration. Notice it is the opposite of diffusion. Diffusion goes from high concentration to low concentration. You do not need energy for that, because it's like if you take a ball and you put it on top of a hill, naturally the ball would roll down the hill. That is what happens in diffusion. But active transport, you're putting the ball at the base of the hill and you want the ball to go up the hill. So you have to use energy to roll the ball up the hill, right? It takes effort. So passive transport, that's diffusion and osmosis, no effort is required. Okay, you just need that gradient, okay, from high to low, right? Diffusion, high concentration of particles to low concentration of particles. Osmosis, high concentration of water to low concentration of water. So you need that gradient for diffusion and osmosis. However, active transport, it doesn't matter the gradient, you just need energy to push the molecules against the concentration gradient from low to high. Active transport allows cells to accumulate high concentration of important substances, example glucose, amino acids, and ions on one side of a membrane. Okay, and just as diffusion and osmosis, active transport is very important to living organisms. So for instance, you need active transport so that minerals can be absorbed from the soil into plant roots. Okay, you need the minerals to go in one direction. When in any living organism, once you need things to go in one direction, you need active transport. You need energy for that to happen, right? You need active transport to transport sugars produced during photosynthesis into the phloem of leaves. Okay, and also some of the glucose and amino acids produced in digestion are absorbed from the ileum into the bloodstream by active transport. Active transport is also needed to reabsorb useful substances from the filtrate in the kidney tubules into the blood. So active transport, just as diffusion and osmosis, is very important. Good. So there are a few points I want you to recall from this lesson. First of all, you need to recall that diffusion and osmosis are examples of passive transport. They do not require energy to occur. They simply rely on the concentration gradients of the molecules being transported. Secondly, active transport requires energy generated during respiration to move materials against the concentration gradients, and that is from an area of lower concentration to an area, and it depends on its concentration in relation to another solution with which it is compared. Now, every solution has a solid concentration, a measure of its proportion of solids, and a water concentration, which is a measure of its proportion of water. And if the water concentration is high, then the solid concentration would be low. And if the water concentration is low, then the solid concentration will be high. Okay. The next thing I want you to remember is that a solution with an extremely high solid concentration is said to be concentrated. One that has a very low solid concentration is said to be dilute, and the percentage of water in a concentrated solution is very low, whereas the percentage water in a dilute solution is very high. Next point, in osmosis, water would always move from a dilute solution to a concentrated solution, but it must be through a differentially permeable membrane. Okay? Cytolysis or bursting occurs in animal cells when too much water enters the membranes by osmosis, and such that they cannot withstand the tug of pressure inside the cell. This does not happen in plant cells because of the rigid cell wall. Okay? Remember that. The last point is that when water leaves a plant cell by osmosis, it becomes plasmolysed, and plasmolysis is when the cell membrane of a plant cell separates from its wall. Okay, so plant cells undergo plasmolysis when they are placed in a hypertonic solution. Okay, animal cells undergo cytolysis when they are placed in a hypotonic or extremely hypotonic solution, right? So keep those things in mind, study the slides carefully. Thank you for paying attention to this video, and please remember to like, comment, and subscribe. Don't forget to hit the bell notification button for more videos like this. Thank you very much. Enjoy the rest of your day.